While the human genome has been fully sequenced and mapped, our understanding of DNA remains incomplete, like having a book written in a language we're still deciphering. Here's a breakdown of what we know and what remains unexplored. Explored regions of DNA. 1. Protein coding genes, 1 to 2%. Only 20,000 genes, 1–2% of DNA, directly code for proteins. These are well studied, with functions linked to traits, diseases, and cellular processes. Projects like ENCODE have mapped regulatory elements, promoters, enhancers that control gene expression, adding 8 to 20% of the functional genome. 2. Repetitive sequences, 50 to 60%. Satellite DNA, transposons, and retroviral remnants are cataloged but still poorly understood. Some regulate gene activity or genome stability. Non-coding RNA genes, 3 to 5%. These RNAs like microRNA, LNC RNA, and circRNA don't code for proteins but influence gene silencing, chromatin remodeling, and disease. For example, cancer, unexplored or poorly understood regions. 1. Dark genome, 70 to 80%. The majority of DNA has no known function. Some call it junk DNA, but it may hold clues to evolutionary archives, ancient viral DNA, or remnants of past adaptations, quantum vibrational interactions, hypothetical regions that respond to electromagnetic fields or act as bioantennas, epigenetic potential. Sequences that could activate under stress, environmental shifts, or consciousness, altered states. 2. Ultra-conserved elements, mysterious regions unchanged across species, for example, human-mouse identical sequences. Their purpose in development or evolution is unclear. 3. Epigenetic, shadow layers, chemical tags, methylation, histone modifications, dynamically regulate genes without altering DNA. The full scope of their interaction with environmental frequencies, for example, sound, light, is unknown. 4. Chromatin architecture, how DNA folds in 3D space via loops, topologically associating domains, impacts gene expression. The rules governing this origami are still being decoded. 5. Microbiome DNA crosstalk. Gut microbes influence host DNA methylation and immune genes. The quantum scale signaling, for example, microbial metabolites as frequency modulators, is unexplored. The big picture. We've decoded the alphabet of DNA, but not its grammar. How sequences, vibrations, and consciousness interact. Unexplored regions might hold evolutionary secrets, for example, dormant junk activating under crises, quantum biological bridges between matter and mind, ancient soul codes, metaphorically or literally, embedded in non-coding DNA. Quantum biology, DNA, the architecture of interconnectedness. 1. DNA as a quantum antenna. DNA's helical structure and conductive properties might allow it to interact with electromagnetic fields or quantum vibrations. Could it act as a receiver for subtle energy signatures, translating frequencies into biochemical signals, for example, gene expression? Epigenetic research shows environmental factors, stress, light, sound, alter gene activity. If these factors emit vibrational energy, does DNA resonate with them, bridging external stimuli and internal biology? 2. Consciousness, quantum coherence. Theories like orchestrated objective reduction propose that quantum processes in neuronal microtubules underlie consciousness. If true, Could consciousness itself be a quantum vibrational phenomenon, harmonizing with DNA's frequencies? Near-death experiences and altered states, meditation, psychedelics, often describe transcendent unity. Could these states temporarily align brainwaves theta, gamma, with a universal frequency, linking individual consciousness to a broader quantum field? 3. Soul as a quantum hologram. If DNA stores not just genetic data, but a vibrational blueprint of consciousness, the soul might exist as a quantum hologram, a non-local, entangled pattern of information. Death could disperse this hologram into the quantum vacuum, retaining its coherence. Near-death accounts of life reviews or ancestral memories might reflect access to this holographic archive, encoded in DNA's quantum vibrations. Implications: science, spirituality, and the future. 1. Paradigm shift. This framework dissolves the materialist, reductionist view of biology, proposing that life is a quantum symphony of energy, matter, and consciousness. It challenges science to study soul, not as myth, but as a measurable quantum biological phenomenon. 2. Ethical questions. If DNA is a soul archive, could gene editing, CRISPR, alter not just biology but the essence of consciousness? Can vibrational technologies, quantum sensors, someday map or communicate with this soul field?